Hello, everybody. Today we're going to be talking about the spinal cord anatomy, and we're going to be mentioning some important anatomical landmarks and their clinical correlation. And we're going to be looking at a cross-section of the spinal cord and analyzing the internal structure of the spinal cord. Now, if you like the content, don't forget to like and subscribe, and you might want to check our membership plans where you can enter private questions room for members and get access to videos where I solve questions that you ask in those private rooms. So we're going to be sending out weekly videos for members where I'm going to be answering their questions. So if you're interested in that, you might want to check it out, like and subscribe, and without further ado, let's get to it. So today we're going to be talking about the spinal cord anatomy. So first of all, our nervous system is divided into CNS, central nervous system, and PNS, peripheral nervous system. So the central nervous system is composed of the brain and the spinal cord. The peripheral nervous system is composed of the spinal nerves and the cranial nerves. A small exception is that the first and second cranial nerves are considered to be a part of the central nervous system. But we're going to be talking about the cranial nerves in later videos, so for this video, just know that the central nervous system is the brain and the spinal cord. The peripheral nervous system is the spinal nerves and the cranial nerves. So this here is a basic overview of the brain. This is the cerebrum, composed of a right and left cerebral hemispheres. They're divided into lobes. We're going to be talking about those in later videos. This is the cerebellum, and this is the brainstem extending into the spinal cord. So this here is the spinal cord housed in the vertebral canal. And the spinal cord gives off spinal nerves. So we have 31 pairs of spinal nerves. They're divided into 8 cervical, 12 thoracic, 5 lumbar, and 5 sacral, and finally 1 coccygeal. So here C1 to C8, 8 cervical. Here 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. So the spinal cord starts up here in the upper border of C1 and it ends down here in the lower border of L1. We have an important point we need to make here is that the spinal nerves at the cervical segment here exit above their respective vertebra, meaning the C4 here, for instance, exits above the C4 vertebra. The C3 spinal nerve exits above the C3. The C6 exits above the C6. So in the cervical segment, the spinal nerve exits above its respective vertebra. In the other segments, the spinal nerve exits below the respective vertebra. So here, the T6 exits below the T6. The T7 exits below the T7. All right? So during the intrauterine life, the bones grow at a more rapid pace than the soft tissue, such as the spinal cord. So that's why the vertebral column here, which is bony, is bigger than the spinal cord, which ends here at the lower border of the L1. So the very last portion of the spinal cord is called the conus medullaris. It corresponds to the lower border of L1 if we're talking about the vertebral column, and if we're talking about the spinal cord segment, it corresponds to S3 to S5, and the portion just above the conus medullaris is called the epiconus. It corresponds to T12 to L1, this portion here, and if we're talking about the segment, the specific segment of the spinal cord, it corresponds to L4 to S2. All right. And down here, this part here, this is basically the lumbosacral nerve roots coming out of the conus and epiconus. So the conus medullaris and epiconus gives off lumbosacral spinal roots and they make this horse tail shape, and we call this the coda equina, which means horse tail in Latin. Now, we want to talk about the lumbar puncture. So what is the lumbar puncture? Basically, sometimes a patient may present with a CNS infection, such as meningitis, and we are called upon to try to find out what caused this CNS infection. So in some instances, we have to insert a needle and take a sample of the cerebrospinal fluid, or CSF, and then analyze it and look at its cells and know what caused this CNS infection, whether it's bacterial, viral, etc. So to do that, we need to insert a needle at a specific point. We need to be aware of the specific point at which we insert the needle, okay? So, first of all, we need to insert the needle at a point where we don't injure the spinal cord. So it has to be below the spinal cord. And we just knew that the spinal cord ends at the lower border of L1. So we want to be below that, all right? And to be more sure, we want to be below it like by a long shot, so we don't risk at all injuring the spinal cord. So basically, we agreed that the best part where we can insert the needle so that we don't injure the spinal cord is between L3 and L5. And there's this stupid mnemonic saying L3, L4, L5, keep the spinal cord alive. So anyways, we can insert it between L3 and L4 or between L4 and L5. Usually what's done is that we insert it at the level between L4 and L5. Now, how do we localize that level? We look at something called the iliac crest here. The highest portion of the iliac crest, if we draw a line to the back, that's the L4 vertebra. And we can insert the needle below that just a little bit. Okay, so this is the iliac crest here in the surface anatomy. Now, as we insert the needle, we pierce certain structures to reach the portion of the spinal, the portion of the CSF that we want to take to analyze. So we need to be aware of the structures that we pierce with the needle, okay? So obviously, first we pierce the skin and the fascia, and then we pierce a couple of ligaments. So the first one here, this ligament, which is above the supraspinal ligament. It's above the spinous process, so it's called the supraspinal ligament because it's above the spinous process. And then we pierce this ligament here between two spinous processes called the interspinal ligament. And then we pierce this here, this ligament called the ligamentum flavum. So first of all, we pierce the skin and the fascia, then the supraspinal ligament, then the interspinous ligament, then the ligamentum flavum. And after piercing the ligamentum flavum, now we are in the meninges. So our target is to reach the subarachnoid space, and this is the space where the CSF resides. So our meninges is basically a dura mater, arachnoid, and pia mater. And between each of those three layers, we have a space. So this first, after the ligamentum flavum, this space here is called the epidural space because it's above the dura. And this golden one is the dura, right? And then after the dura, there's a subdural space, and then the arachnoid, and then the subarachnoid space. So the subarachnoid space is the space between the arachnoid and the pia mater. So this is the layer where we take the CSF from. So first of all, the skin and the fascia, then the supraspinous ligament, then the interspinous ligament, then the ligamentum flavum, then the epidural space, the dura, the subdural space, arachnoid, and then the subarachnoid. All right. So that's all we have to talk about when it comes to the lumbar puncture, okay? So, before we can continue, we need to have a basic overview of the anatomy of the neuron. So the neuron is basically the basic cell of the nervous system. is composed of these little spikes here called dendrites. These receive impulse, and then this impulse is transmitted along this long axon into the axon terminals, and those axons are lined by something called the myelin sheath, which helps to speed up the conduction of the impulse, and we're going to be talking about those later. And in the center here we have this part, which is called the cell body or soma, and this is the site of protein and neurotransmitter synthesis. Okay, so a group of cell bodies in the CNS is called nucleus or column, depending on its shape. So a group of cell bodies in the CNS, if it's circular, it's called nucleus. If it's in a columnar shape, it's called column. And a group of cell body in the peripheral nervous system is called ganglion. Okay, and a group of axons is called tract or fasciculi. Okay, so now we need to take a cut section of the spinal cord and look at it and analyze it. So this is a cut section of the spinal cord. This portion here is the white matter, composed of many tracts. So as we said before, a group of axons is called a tract. So this part here, called the white matter, is composed of tracts, and this butterfly edge-shaped portion in the center is the gray matter. It's composed of cell bodies. So this portion here of the gray matter is the ventral horn, and this portion here is the dorsal horn, and the dorsal horn gives off a dorsal root, and the ventral horn gives off a ventral root, and then the dorsal and the ventral root join here to create a spinal nerve, and then this spinal nerve here gives off a dorsal and a ventral rami. Okay, so the dorsal horn is basically responsible for sensations. So as we can see here, if we, for instance, touch an object, the impulse goes from the hands all the way to the dorsal horn, and then from the dorsal horn up the spinal cord to the brain. And if you want to move a muscle, it goes through the ventral horn. So the ventral horns are responsible for movement and motor function. So it goes from the brain to the ventral horn, to the spinal nerve, to the muscle. Okay? So what else do we need to talk about at the cut section? There's something called the lateral horn that's present in some spinal cord segments, not all of them. It's present from the level of T1 to L2, and it corresponds to preganglionic sympathetic neurons, and we're going to be talking about those in later videos. Okay. Now, one thing more we need to know about the white matter. As we said, it's composed of tracts. We have two types of tracts, ascending and descending tracts. And we're going to be talking about those in later videos, but just know that the descending tracts is called descending because they go from the brain to the spinal cord to the muscle. So those are the motor tracts. They go from the brain to the spinal cord to the ventral horn to the muscle. And we have the ascending tracts, which are the sensory tracts. They go from the object of sensation, say for instance the finger, the touching, and then to the dorsal horn, up the spinal cord to the brain, and that's why they are called the ascending tracts. Okay? So the ventral horns are composed of motor neurons, as we said before, and those motor neurons end up innervating the muscle. So those motor neurons are composed of two types, alpha motor neurons and gamma motor neurons. The alpha motor neurons are responsible for muscle contraction, and the gamma motor neurons are responsible for sensation of changes in tension of the muscle. So for instance, if we do a knee reflex and we hit with a hammer here at the tendon, there's a change, there's an increase of tension on that muscle. What senses that increase in tension is the gamma motor neurons. Okay, so the alpha motor neurons are responsible for muscle contraction. The gamma motor neurons are reacts to changes in muscle tension. Okay. So the alpha and gamma motor neurons are arranged in the ventral horn. Some of them are located medially near the medial side here, and some of them are located laterally here, and some of them are located ventrally here, and some of them are located dorsally here. Okay, so the motor neurons that are located dorsally here are responsible for innervating the flexor muscles, such as the biceps, if we're talking about the upper limb. And the ventral motor neurons here are responsible for innervating the extensor muscles, such as the triceps, if we're talking about the upper limb. And the motor neurons in the medial side are responsible for innervating the proximal structures of our body. And on the lateral side, the motor neurons are responsible for innervating the distal structures of our body. So a quick microbiology integration here. So those motor neurons innervate the muscles, as we said before, but they have an inhibitory feedback mechanism, and it's designed so that those motor neurons don't overactivate and end up causing muscle spasms and excessive contraction. So they need some form of inhibition. There are a couple of ways by which they're inhibited. The one we're going to be talking about now is this way here, through an inhibitory interneuron. So there's this inhibitory interneuron called the Renshaw cell. It releases inhibitory neurotransmitters such as GABA and glycine, and we're going to be talking about neurotransmitters in later videos. And those neurotransmitters prevent this motor neuron from firing excessively and leading to muscle spasms. So there's a bacteria called the Clostridium tetani. It releases a neurotoxin called the tetanospasmine, and this tetanospasmine ends up damaging the Renshaw cell. So when it damages the Renshaw cell, the motor neurons lose the inhibition feedback mechanism, and they end up firing excessively and causing spasms and causing a condition called a tetanus, which can present with spasms, a lockjaw or rhizus sardonicus, and opisthotonus, or the excessive arching of the back, such as here. This here is the lockjaw or rhizus sardonicus. The patient looks like he has a sort of a smile on his face. And the opisthotonus is the excessive arching of the back due to excessive spasm of the back muscles. Okay, so the last thing we want to talk about in this lecture is that we want to be able to differentiate between the different spinal segments. So we want to know, if we're looking at a spinal segment, we want to know whether it's the cervical or thoracic or the lumbar or the sacral. So we differentiate, forget about the extra details, we just want to differentiate between those segments via 4 factors. So the first factor is the white to gray matter from lumbar to cervical, meaning from the lumbar or even from the sacral. So from down up, we want to compare the ratio of the white matter here to the gray matter, okay? And the second factor is the dorsal column. So these parts here are called the dorsal columns. We're going to be talking about them in later videos. So as you can see, in certain segments, they are increased. So we have 2 dorsal columns here and 2 here. But down here, we just have 1 and 1, right? So we want to differentiate between the dorsal columns in the different spinal segments. And we want to differentiate between them according to the ventral horn enlargement. So as you can see, there are some spinal segments with a normal ventral horn, but some have an enlarged ventral horn, such as here and here. And the last factor is the lateral horn presence. Some spinal segments have lateral horns and some don't. So let's start with the first factor, white to gray matter. So as you can see here, if we go from down up, the white matter starts increasing, as you can see here. So why is that? Well, because as we said before, the white matter is composed of tracts, which are a group of axons. And when we are in the lower spinal segments, those group of axons are responsible for innervating the lower limbs only. All right, as we go up, those axons start to innervate the lower limbs and the upper limbs as well. So now we have more tracts, and that's why as we go from down up, we have more white matter. Okay, so the first factor, white to gray matter from down up, the white matter increases, okay? The second factor is the dorsal column. The dorsal columns, and we're going to be talking about those in later videos, but they play a role in certain sensations such as the pressure and vibration, etc. And we basically have two types of dorsal columns. So we have this medial one called the gracile tract, and we have the lateral one called the cuneate tract, okay? The gracile tract is responsible for the sensations from the lower limb, and the cuneate tract is responsible for the sensations of the upper limb. So if we divide the spinal cord to a portion above T7 and below T7, what we can see is that the spinal segments below the T7 are composed of only one dorsal column here and one dorsal column here. But above the T7, we have two dorsal columns here and two dorsal columns here. So why is that? Well, because above T7, we have dorsal columns innervating the lower limb, i.e. the gracile tracts, and we have other dorsal columns innervating the upper limb, the cuneate tract, right? But below T7, we are now below the level of the upper limbs, so that's why we only have the dorsal column innervating the lower limb, which is the gracile tract. So to recap, below T7, we only have the gracile tract, and above T7, we have the gracile tract and the cuneate tract, okay? So again, above T7, the gracile tract. That's actually, that's a typo, okay? So above T7, we have both the gracile and the cuneate tract, and below T7, we have the gracile tract only, okay? So the third factor is the ventral horn enlargement. There are two portions of the spinal cord where we have a ventral horn enlargement. So as we said before, the ventral horn is responsible for the motor innervation. So the portions of the spinal cord innervating the upper limb muscles and the lower limb muscles, which are two portions of our bodies that have like an increased amount of muscles. So those spinal segments should have an enlarged ventral horn to be able to innervate those large muscles, right? So about the upper limb, so for the upper limb, we have a ventral horn enlargement at the cervical level. So there's a ventral horn enlargement at the C5 to T1 level. And this is the level of the brachial plexus innervating the upper limbs. And we have a similar ventral horn enlargement in the lower limb corresponding to the lumbosacral plexus innervating the lower limbs at the level of L2 to S2. So we have two portions of the spinal cord with ventral horn enlargements. One corresponding to the brachial plexus innervating the upper limb, which is C5 to T1, and one corresponding to the lumbosacral plexus innervating the lower limbs from L2 to S2. And finally, the lateral horn, and we mentioned that in a previous slide, is basically corresponds basically to the sympathetic preganglionic neuron, and we're going to be talking about that in later videos. And this is basically from the level of T1 to L2. So that's it for this lecture. I hope it was useful, and if you like the content, don't forget to like and subscribe. If you have any questions, you can put them in the comments. If you want to check out the membership plans, feel free to do so, and I'll see you in the next lecture.